We are heading straight into the field today, into the dense, humid rainforests of the Western Ghats of India.
Hmm.
If you're looking for a global hotspot of biological discovery, I mean, this is it.
It really is.
But sometimes the thrill of finding a new species isn't about trekking to some untouched peak. Sometimes it's about, looking closer at what you thought you already knew.
That's the perfect frame for this deep dive. Our subject today is the Sahyadri spotted flitter, now formerly known as Zographetus mathewi.
Right.
And this story is, well, it's a magnificent testament to painstaking scientific rigor. It required a team of dedicated researchers to peer past the obvious and find a creature basically hiding in plain sight.
Our mission today is simple. We are taking the sources the team provided, the morphological papers, the ecology notes, the conservation reports, and we're going to understand how they definitively proved this butterfly was a new species.
And maybe more importantly,
And more importantly, why its discovery in the highly pressured, low-elevation forests of Kerala forces this critical reassessment of conservation strategy for the whole region.
to start, this is a globally relevant finding. This new flitter, it's the 15th known species in the genus Zographetus.
15th.
And for India specifically, it's only the 5th one ever recorded. This effort wasn't isolated either. It's the result of a strong collaboration
Between who?
between Dr. Kailesh Sadasivan of the Travancore Nature History Society, researchers from the Zoological Survey of India, and INTROC, the Institute of Tropical Research, Ecology, and Conservation. It took multiple institutions to confirm all the lines of evidence.
So stepping back from the official paperwork, the story of this discovery really starts with an identity crisis.
Yes.
When the first specimens were collected in these low-elevation evergreen forests in Kerala, they were immediately flagged as something familiar.
Right. They thought it was the common and widespread purple-spotted flitter, Zographetus ogygia. And that misidentification is key to understanding what makes this butterfly so special. Many closely related insects are what we call cryptic species,
Cryptic meaning?
meaning they look so similar to existing species that you can't reliably tell them apart just by, casual observation in the field. They are hiding in plain sight.
So if the initial visual check said, this is Z. ogygia, what made the researchers suddenly pump the brakes? What was that first subtle clue that suggested, wait a minute, maybe this is something else?
Well, the team noticed these slight inconsistencies in the wing patterns, specifically in the placement of the dark spots. They just didn't perfectly align with the known variations of Zoogygia. In taxonomy, that kind of slight difference is often the flicker of a match that leads to a fire.
And when you're dealing with a cryptic species, you have to raise the bar for proof.
Oh, significantly.
Mhm.
The source material is very clear on this. To move past that initial doubt, they had to conduct what they called rigorous morphological analysis and genital dissection.
Wow. So this isn't just counting spots. This is deconstructing the creature itself.
Absolutely. And that confirmed they had a distinct lineage, separate from Zoogygia. Once the proof was irrefutable, the findings were published in the peer-reviewed journal Entomon.
And I really appreciate how they approached the naming. The common name, the Sahyadri spotted flitter, is beautiful.
It is.
It roots the species right in its home, referencing Sahyadri, which is the local name for the Western Ghats mountain range itself. It immediately tells you it's an endemic species.
And the scientific name mathewi carries deep significance too. It's an homage to Dr. George Matthew.
A distinguished Indian entomologist.
A truly distinguished one, yeah. He spent his career focused on forest entomology in the Western Ghats, and his work created the baseline knowledge that let modern researchers even spot these subtle anomalies. It's a tribute that connects past science with current discovery.
They also had to place it systematically, right? So this new flitter belongs to the family Hesperiidae, the skippers,
Right.
and specifically it was assigned to the Zographetus satwa group. Now, this is where we have to wade into some technical waters, but it's important for understanding the proof.
Let's do it. The distinction between the Z Satwa group and the Z Ogygia group relies on complex wing venation.
Venation, so the veins in the wing.
Exactly. Imagine the butterfly's wing as a road map. The researchers are looking at the specific location of the highway junctions. The diagnostic feature here is how vein C2 arises significantly closer to the wing base than vein C1.
Okay, so we're talking about where two specific highways diverge. The precise distance between their origins is like a genetic roadmap.
Precisely. That detail is genetically reliable, unlike, say, the intensity of a color spot, which can vary.
This is where it gets really interesting, though, stepping away from the venation and back to what you can see. If they look so similar, what was the first striking, visible clue?
Ah, yes. The sources call it the ochre clue.
The ochre clue.
If you turn the butterfly over and look at the underside of the hind wings, the difference is unmistakable. Where its close relative, Zogaphetus, is this uniform, deep chocolate brown. Z. mathewi flashes a distinct, bright, yellow ochre scaling. It's like this vibrant splash of color on an otherwise muted background.
That sounds like a gift for field researchers, but you're saying even with that ochre scaling, identification is still notoriously difficult.
It is. It's still a cryptic species, and the males in particular hold secrets you could only see up close.
What kind of secrets?
They have specific secondary sexual characteristics that function as reproductive markers. There are two main tiny structures. First, the basal portion of that vein, remember that highway along with the cubitus vein, is noticeably swollen.
Just a subtle thickening?
A very subtle thickening, yeah. And the second one is that the male has a thick tuft of specialized, long black hairs right at the base of the forewing underside.
And we're talking about structures you'd need a microscope to see.
Oh, a high-powered microscope, yeah. You're trying to spot a difference of a few microns. It really makes you appreciate the incredible patience these scientists have.
It's astonishing precision, but as we said, even those tiny hair tufts can be misleading. The sources make it clear that the ultimate decider for these cryptic flitters is the internal anatomy.
That's right. Because in insect evolution, the structures for reproduction evolve very quickly. This genital morphology acts as the definitive biological lock and key.
The lock and key.
If the structures don't interlock perfectly, the species can't successfully interbreed. That instantly confirms they are separate species.
So let's go through this comparison for the listener. What are the precise anatomical variations they found between Z. mathewi and its doppelganger, Z. ogygia?
Okay, so we're comparing three critical components. Think of the male genitalia as having a main gripping mechanism. That's the valva, and a specialized latch, the uncus.
A grip and a latch, got it.
In Z. mathewi, the uncus, the latch, is long and thin, and it has this distinct little window or opening called a fenestrula.
A fenestrula.
Okay. Now contrast that with Z. ogygia, where the uncus is broader and completely lacks that window.
So a completely different structure.
A completely different structure. Now look at the valva, the main grip. In the new species, its dorsal margin is shallowly convex at the base, and the end portion is noticeably constricted. Z. ogygia has a much more uniform, simple valva.
So they aren't just slightly different, they have fundamentally different architecture. It proves millennia of separate evolution.
Exactly. And the final piece is the cecus, which is part of the support structure. In Z. mathewi, it's relatively short and bent slightly upward, whereas in Z. ogygia, it's longer and straighter.
Wow.
These differences, the fenestrula, the constriction, the curve, are the definitive microscopic proofs.
Moving from that incredible lab detail back out into the jungle, the next big breakthrough was about the butterfly's dinner. The sources confirm that the larvae of Z. mathewi are strictly monophagous.
The term is crucial. It means they are specialist feeders. They feed exclusively on one specific plant, Aganope thursiflora.
Which is a kind of...
It's a tough, woody climber, sort of a shrub, in the pea family. It's not a generalist. It requires this one specific host.
And finding the larvae on this specific plant allowed the researchers to close the final loop on the identification, right?
Yes, that was the ecological linchpin. By identifying the exact host plant, they could collect larvae and pupa and actually rear the species in captivity.
Oh, clever.
Observing the entire life cycle, from larva to the adult flitter, confirmed the whole identity chain. It connected the habitat, the diet, and the unique adult anatomy.
But despite confirming its life cycle, there's this captivating mystery surrounding the adult flitter, which the sources call the elusive adult phenomenon.
It's a great name for it.
Why do we know so much about the larva but so little about the butterfly itself?
The statistics are truly staggering. Despite years of intensive, targeted surveys across its known habitat, only a single adult specimen has ever been formally documented in the wild.
One. Just one. That sounds like a species teetering on the brink of extinction.
It's counterintuitive, isn't it? But the species is not considered rare or unstable.
How can that be?
The researchers found plenty of evidence of it. Larvae, pupa, feeding damage on the host plants, all were frequent across its known locations. This suggests the population is actually robust and stable in its small endemic habitat.
So why is the adult never seen?
It comes down to behavior. The assumption is the adults are either strictly canopy-dwelling,
Meaning they live high up in the trees.
exactly, high up in the dense tree cover where we rarely look, or, and this is perhaps more likely, they have a very, very short flight period.
How short are we talking?
Researchers think they might only be flying actively for, say, 2 weeks a year, maybe just a few hours a day. That makes the window for detection incredibly small. It's a species that actively avoids being observed.
And this highly secretive, specialized, yet stable species is confined strictly to one very specific environment. Where exactly is its home range?
It's strictly endemic to the low-elevation evergreen forests of the Western Ghats, and the altitude limit is critical, below 600 meters above sea level.
Below 600 meters. Okay.
Confirmed records exist in biodiversity hotspots like Kalar, Shendurni Wildlife Sanctuary, Adamalair, and Nilamber.
How does the fact that this species is found exclusively below 600 meters change how conservationists have to think about the Western Ghats? That altitude detail seems to be the core of the conservation story here.
It highlights a critical blind spot. Historically, conservation efforts across the Western Ghats have focused immense resources on the high-altitude, visually stunning ecosystems.
Right, the Shola grasslands.
Exactly. Those areas are charismatic. They have iconic large mammals, stunning landscapes that naturally attracts funding and public interest.
But that means everything below those majestic peaks gets less scrutiny.
Precisely.
Yeah.
The discovery of Z. mathewi forces this critical shift, reminding us that the lowlands, those forests below 600 meters, are vital genetic reservoirs for endemic insects and other unique life that can't survive anywhere else.
And these low-elevation corridors are precisely the areas under the most immediate, highest pressure. They're fragmented, they're vulnerable, and they're constantly shrinking.
They are. I mean, if you look at the geography, these low-elevation areas are where human activity most aggressively intersects with the natural forest.
Like what?
We're talking about corridors being choked off by agriculture, rubber, or spice plantations, rapid urbanization, new highways, resort development for tourism.
Yeah.
Imagine a small patch of forest isolated between a highway and a new resort. The destruction of that patch means genetic isolation for the flitter, likely leading to local extinction.
So protecting a species like the Sahyadri spotted flitter is less about grand national parks and more about preserving these fragmented, overlooked, low-altitude forest patches.
It requires a more nuanced, site-specific strategy that addresses fragmentation directly. We have to recognize these lowlands as essential genetic banks, not just buffer zones that can be sacrificed. This tiny butterfly is now the emblem for an entire threatened ecosystem.
This has been a fascinating journey. From noticing a flicker of yellow ochre on a hindwing, to the incredible anatomical proof from the biological lock and key, to understanding the secretive nature of an adult butterfly that is stable yet almost never seen. We've established the Sahyadri spotted flitter is a specialist, endemic to low-altitude forests, and its existence highlights a dangerous blind spot in conservation.
What's fascinating here is how often the smallest, most easily overlooked creature serves as the canary in the coal mine for the health of an entire habitat.
Yeah.
And given that a species as unique as the Sahyadri spotted flitter went unnoticed for so long in these overlooked, highly threatened low-altitude forests, this raises an important question for you, the listener. How many other vital endemic species might currently be silently struggling, unknown to science, in the most fragmented and pressured pockets of the Western Ghats?